Hello, Bio 220 students. Welcome once again to Online Anatomy. Chapter 4 is going to deal with functional anatomy of prokaryotic and eukaryotic cells. So, cells that do not and do have organelles. So, prokaryotes, well, it is from Greek, which means pre-nucleus. So it's giving a hint that prokaryotic cells, they do not have individual organelles where eukaryotes do. In particular, eukaryotes have a nucleus. Some defining things about a prokaryote is that one circular chromosome, as far as the genetic material is concerned, and it's not in a membrane. There are no histones to wrap around that genetic material. There are no individual organelles in prokaryotes. Bacteria are a type of prokaryote that have a peptidoglycan cell walls, whereas archaea have pseudomerin cell walls. And, well, they reproduce by binary fission. Eukaryotes, they have paired chromosomes in a nuclear membrane, so they are wrapped. And histones are used to help pull that genetic material tightly packed. Eukaryotes have organelles that have defined functions in specific parts of the cell, as opposed to just doing all what a prokaryote does in any part of its cell. The cell walls, if they are present, like in the case of plants, are made out of polysaccharides, and they divide by a process called mitosis instead of traditional prokaryotic binary fission. As far as the size, shape, and arrangement of bacterial cells, well, typically bacteria can range from 0.2 to 2 micrometers in diameter and about 2 to 8 micrometers in length, where most bacteria are monomorphic, which means they have one particular shape for that type of bacteria. There are a few pleomorphic bacteria that can have many different shapes. Now, getting into some of the shapes, the more common ones are rod-shaped bacteria like Bacillus, spherical-shaped bacteria like Coccus. There's also spiral-shaped bacteria like Vibrio spirillum or Spirochaete. There are star-shaped bacteria, and there are also those that are rectangular. Now, going further, sometimes they're arranged. Bacterial cells will sometimes be arranged in pairs like Diplococci or Diplobacilli. There are also certain type of bacteria that like to form clusters like Staphylococci, and sometimes they'll form chains with each other like Streptococci, cocci, and Streptobacilli. Sometimes bacteria will in groups of 4 or tetrads, and then there are examples where they actually form cube-like groups of 8. An example would be Sarcenae. Now, as far as the scientific name, well, we can look at Bacillus, which has the shape Bacillus. So, structure of a prokaryotic cell. Well, you're not going to see individual organelles. More or less, there will be ribosomes, and there will be certain structures like flagellum that help it move around, as well as cilia to help it sense its environment. Now, one example of an external structure bacteria will have are glycocoxylics. They are on the outside of the cell wall. They're very viscous and gelatinous, and made out of polysaccharides or polypeptides, so numerous carbohydrates, sugars, or numerous amino acid proteins. Two types include capsules, which are neatly organized and firmly attached, also slime layer, which is an unorganized or loose form of glycocalyx. Now, these actually contribute to virulence, or how infectious these bacteria can be. The capsules essentially prevent phagocytosis, prevent white blood cells from engulfing, and extracellular polymeric substances. They help form biofilms, which can protect bacteria from removal or help firmly cement them to surfaces, like on our teeth. Now, another structure would be the flagella. These are filamentous appendages that are external to the cell, and these really help move the cell. They're the reason why bacteria are motile, M-O-T-I-L-E. They're able to move on their own. They propel the bacteria, and they're made out of a protein substance called flagellin. There are essentially three parts to flagella, which is the filament. It's the outermost region of that propeller. It has a hook that helps attach the filament to the cell. And then there's the basal body, which consists of rod and pairs of rings. And, well, it anchors the flagellum to the cell wall and cell membrane. So, once again, gram-positive, they'll have very thick cell walls, whereas gram-negative bacteria will have either a different lipid bilayer or a thinner cell wall in general. Now, continuing with flagella, they allow bacteria to move toward or away from stimuli. Now, these are called taxes. Essentially, it might be some nutrient that the cell will want, or it might be some threatening toxin that the cell should get away from. Flagella rotate to basically run or tumble. That's essentially how bacterial cells move. And flagella proteins are H antigens and distinguish among serovars. One example would be E. coli. Now, motility, which is essentially the ability to move on its own to cause its own propulsion. Flagella and bacterial motility. Bacterium, they move basically through running and tumbling. Notice that the direction of flagellar rotation, the blue arrows, determines which of these movements occurs. Gray arrows indicate direction of movement of the microbe. Flagella and bacterial motility. A Proteus cell in the swarming stage may have more than 1,000 peritrichous flagella extending outward. Now, there is an additional structure called the axial filaments, and some textbooks will call them the endoflagella. They're found in Spirochetes, anchored at one end of a cell, and the rotation causes the cell to move in a corkscrew fashion. Axial filaments, well, this is just a diagram that shows that they wrap around parts of a spirochete. Fimbriae are hair-like appendages that allow bacteria to attach to surfaces. And they'll be found all around the cell. And then also pili. They assist in motility in the movement of the bacteria, essentially gliding and twitching. Conjugation pili, they're involved in DNA transfer. Essentially, when they attach from one bacteria to another, it allows for the transfer of DNA from the first bacteria to a second. The cell wall. Now, this essentially prevents osmotic lysis and protects the cell membrane. Osmotic lysis essentially being ripped apart through osmotic pressure. Now, these are made of peptidoglycans in the case of bacteria, and they contribute to pathogenicity, how infectious they are. Now, as far as composition characteristics, the peptidoglycan, it's a polymer. It's a sheath mixture of repeating disaccharides in rows. And those two are N-acetylglycosamine, NAG, and N-acetylmuramic acid, NAM. And they show up as rows linked by polypeptides, by proteins. As far as N-acetylglucosamine, NAG, and N-acetylmuramic acid, NAM, this is how they look as when joined by a peptidoglycan series. Now, gram-positive cell walls. Well, once again, they're characteristic by having a thick peptidoglycan cell wall, as well as having teichoic acids. Gram-negative cell walls. Well, they'll have a much thinner peptidoglycan cell wall. They'll have an outer membrane as well to basically substitute for lack of a thick cell wall. And they'll have periplasm space between the outer cell membrane and inner cell membrane. As far as for gram-positive cell walls, teichoic acids, they're lipoteichoic acids, fatty acids linked cell walls to plasma membranes. Now, a wall teichoic acid links the peptidoglycan that consists of the cell wall, and then they will carry a negative charge. This essentially causes a potential that helps regulate the movement of positive ions, cations. Polysaccharides also and teichoic acids provide antigenic specificity. Now, with gram-negative cell walls, the periplasm between the outer membrane and the plasma membrane, or the innermost membrane, contain peptidoglycan, but just not in as thick an amount as gram-positive cell walls. The outer membrane, though, is made of a polysaccharide, lipoproteins, and phospholipids. So, polysaccharides, which are long chains of sugar molecules, carbohydrates, lipoproteins, which are molecules that have a little bit of protein and fat substances, and phospholipids, fatty substances or fats with phosphate groups. Gram-negative cell walls, furthermore, they protect against phagocytes, complements, and antibiotics. They basically help the gram-negative cell stay alive. And they're made of lipopolysaccharides, where O polysaccharide functions as an antigen. Lipid A is an endotoxin embedded in the top layer. There are porins, which are proteins, that form openings or channels through the membrane that allows different substances to go through. In some cases, those porins could be water, or also allowing cations nutrients, certain things to smaller things to go through the cell. Now, cell walls and the Gram stain mechanism. Again, this involves the crystal violet and iodine crystals to form inside the cell. When it comes to gram-positive cells, bacteria, alcohol will dehydrate the peptidoglycan layer. And then CVIs, crystals, crystal violet, Indian crystals will not be able to leave. Whereas with gram-negative cells, alcohol will dissolve the outer membrane and leaves holes in the peptidoglycan layer. Crystal violet iodine crystals will wash out, and those cells are colorless. When you add safranin, is added in order to stain those cells. For gram-positive cell walls, there are two rings in the basal body of the flagella. They produce exotoxins, and they have a high susceptibility to penicillin. So in gram-positive illnesses by gram-positive bacteria, penicillin has been known to be highly effective. Now, essentially, they're just disrupted by lysozymes, which help digest those bacterial cell walls. In the case of gram-negative cell walls, there are four rings in the basal body of the flagella. They produce endotoxins as well as exotoxins, and they have a low susceptibility to penicillin. Atypical cell walls. These could be acid-fast cell walls, like gram-positive cell walls, or those that are made of waxy lipid mycolic acid bound to the peptidoglycan layer. There are also Mycobacterium, Nocardia, and those that can be stained with carbofulcin. As far as atypical cell walls continued, there are mycoplasms. These essentially lack cell walls, and, sterols are present in the plasma membrane, form of alcohol molecule. There are archaea, which are wall-less, or walls are made out of pseudomirin, which essentially lack NAM and D amino acids. So it gives rise to why archaea have a large range of membrane compositions. Now, when the cell wall is damaged, this could be due to a lysozyme that hydrolyzes or breaks apart the bonds between the peptoglycan sheets. There is also penicillin, which inhibits peptide bridging within the peptidoglycan. There are protoplasts, or I should say protoplast is a wall-less gram-positive cell. Spheroplasts would be a wall-less gram-negative cell. Protoplasts and spheroplasts are susceptible to osmotic lysis, essentially after that cell wall has been removed. And then there are L forms, which are wall-less cells that swell into highly irregular shapes. Structures internal to the cell wall. We'll start getting into those. For example, the plasma or cytoplasmic membrane. All cells have this. Essentially, it consists of a phospholipid bilayer that encloses the cytoplasm, the internal liquid material of a cell. Peripheral proteins on the membrane surface are littered throughout, and these are integral and transmembrane proteins penetrate through those membranes. Now, as far as structure, the actual structure of a cell membrane, follows what's called a fluid mosaic model, which essentially it consists of pieces that are able to travel along that plasma membrane is because the plasma membrane is essentially it's viscous as olive oil. The proteins that are embedded inside that plasma membrane are able to move freely for various functions that are needed, as well as the phospholipids can rotate and move laterally, essentially allowing, sort of like ball bearings, allowing the proteins in the cell membrane to glide through. But because half of each phospholipid is drawn to water and half is repelled by water, they form self-sealing layers that help prevent the cytoplasm from leaking out. As far as the functions, the plasma membrane provides selective permeability. It allows the passage of certain molecules, but not everything. They contain enzymes that allow for ATP production, which is adenosine triphosphate is used for energy, or a source of energy for many functions. And then some membranes have photosynthetic pigments on foldings called chromatophores. As far as functions, damage to the membrane by alcohols or quaternary ammonium detergents, as well as polymyxin antibiotics, will cause openings in the cell membrane that will allow leakage of the internal cell contents. Now, as far as movement of materials across that cell membrane or plasma membrane, there is passive processes which do not rely on energy from the cell. They occur naturally without being forced. Essentially, this is through osmosis, where substances on one side of the plasma membrane that are in a high concentration move across the membrane to the other side where it is lower in concentration. And again, this is passive, which means no energy is used up by the cell. But there are cases where the cell has to push things against that gradient of concentration, and these are active processes, where substances are moved from a side of the plasma membrane that has a low concentration of that substance to the other side where the concentration is higher, and energy is needed to perform these processes. Passive process, well, it's simple diffusion. That's the name of the term where something moves from a high concentration easily and naturally to an area of low concentration. And it continues until the concentration of that substance is equal on both sides of the membrane. Now, there is another type of passive process, which is a facilitated diffusion. Again, it requires no energy on the cell itself, but facilitated diffusion involves solute that combines with a transporter protein in the membrane of the cell. These transporter proteins then transport ions and larger molecules across the membrane. So essentially things that would not naturally pass through, these essentially help them to move across the cell, but while still following the concentration gradient, therefore not expending energy. These transporter proteins are going to be essentially natural openings or natural gateways in the membrane. As far as facilitated diffusion through a nonspecific transporter, these just allow things to go through following the concentration gradient, whereas facilitated diffusion through a specific transporter, it will only take in a certain substance and then close off and allow that substance to move through. Now, the whole process between passive processes is osmosis. It's effectively the natural movement of water across a selectively permeable membrane. from where an area of water is in high concentration to where there's a lower concentration of water. It goes through the lipid layer, and it is able to do this because of water channels called aquaporins, where the lipid bilayer consists of parts that do not want to interact with water. These aquaporins act as channels that allow the free flow of water in and out, or out and in. The pressure behind this is called osmotic pressure, which is just the pressure needed to stop the movement of water across the membrane. Here we just have a simple lab activity where essentially there's a substance that's in high concentration inside the bag, and effectively there's very little water inside that semi-permeable membrane. Now the salt, or whatever substance, in this case sugar molecule, that is trapped inside the bag, it can't get out to an area of lower concentration. Therefore, to compensate, water will actually travel into the bag because water is more concentrated on the outside and less concentrated in the bag that's filled with sucrose sugar. As far as these passive processes, there are 3 types of solutions that form, and these have to deal with how much concentration of a dissolved substance is inside or outside of that membrane. For an isotonic solution, essentially the solute or dissolved concentration is equal inside the cell as it is outside the cell. Water is at equilibrium and will not flow. In a hypotonic solution, the outside solution, basically the concentration of that solute, of that salt, sugar, whatever, is much lower outside the cell than inside the cell. Therefore, since those solutes can't easily move into the cell, water will compensate by moving out of the cell, which can cause cells to actually dry up and shrivel. And then there are the reverse case, which is a hypertonic solution, where the concentration of solute is significantly higher outside the cell than inside the cell. And this causes water to move out of the cell. I should correct what I say. Hypotonic means that water will move into the cell, and thus it can cause those cells to actually expand and even burst. Hypertonic solutions are what cause water to move out of the cell and potentially shrivel up and die. Now getting into processes that require energy, or active processes. These involve active transport, which involves a transporter protein that uses adenosine triphosphate, ATP, and it forces things to go against their concentration gradient from an area of low concentration to higher concentration. There is a situation of group translocation, which requires a transporter protein and phosphoenolpyruvic acid, or PEP. It's a substance altered. The substance is altered as it crosses the membrane. Now, as far as the internal liquid of a cell, the cytoplasm, this is what is inside of the plasma membrane. It's the internal volume of a cell. About 8% is water, plus various proteins, carbohydrates, lipids, as well as whatever. And there is a cytoskeleton that acts as a structure to keep things in place. Now, the nucleolid, basically it's a bacterial chromosome, and it has circular thread of DNA. Instead of our double helix in humans, it's a circular piece of DNA that contains the cell's genetic information. There are plasmids, which are extrachromal genetic elements that carry non-crucial genes such as antibiotic resistance, or help produce certain toxins against rival cells. Something that bacterial cells all have is ribosomes, and these are essentially the places where proteins are created through the stringing of multiple amino acids. Ribosomes, they consist of two parts. They're made of protein themselves as well as ribosomal ribonucleic acid. Now, inclusions. Well, there are quite a variety. There are metachromic granules or volutin, which consist of phosphate reserves. There are polysaccharide granules, which are energy reserves. They're filled with carbohydrates. There are lipid inclusions, again used for energy. Sulfur granules for energy. There are carboxysomes, such as rubisco enzyme for the fixation of carbon dioxide during photosynthesis, essentially converting carbon dioxide and water into sugar molecules. And then there are gas vacuoles, which are protein-covered cylinders that help with buoyancy, and magnetosomes. They have iron oxide inside them, and they help destroy hydrogen peroxide, which can do serious damage if left unchecked. Endospores are resting cells produced when nutrients are depleted. They're basically cells turn into endospores when they have no surrounding nutrients. These endospores, or these changed cells, are very resistant to desiccation, to heat, to chemicals, to radiation, desiccation being dried by salts. They can be produced by a variety of bacteria like Bacillus and Clostridium. Sporulation is when the endospore forms, and germination is when the endospore returns to a vegetative state. Essentially forming inside the cell. Eukaryotic cells. Eukaryotic cells have very well-defined structures that have specific purposes. Now, as far as a plant cell goes, they'll have three unique structures, which is a vacuole that contains water. They will have a cell wall, and they will have chloroplasts, which are the site of photosynthesis. Unique to animal cells are centrosomes, centrioles, and pericentriole material, which help provide structure. There are also lysosomes, which help to move and dissolve things, basal bodies, and then flagellum. Let's see. Nucleus, which would be the brain of a cell. Vacuole, which acts as a container of a substance. Mitochondrion, which is where ATP, the energy molecule, is produced. Chloroplasts, so we're looking at a plant cell. Site of photosynthesis, as well as the presence of a cell wall. Now when it comes to flagella and cilia, there is differences between prokaryotic and eukaryotic flagella. Again, these are projections that are used for the movement of the cell or moving substances along the surface. Now, flagella, they're long projections, and they are not as plentiful as cilia, which are much shorter projections. Typically, when we're talking about eukaryotic cells, things are in larger proportion than on prokaryotic cells, which are much smaller. Now, flagella and cilia, they both consist of microtubules, which are made of the protein tubulin. Microtubules are organized as 9 pairs in a ring, plus 2 microtubules in the center. These allow flagella to move in a wave-like manner. Now, continuing on to the cell wall and glycocalyx. Cell wall, it's very unique to plants, algae, and fungi. This is not going to be something in animal cells. And it's made out of carbohydrates. In plants, it's made out of cellulose. In fungi, it's made out of chitin. And in yeast, it will be made out of glucan and mannan. Glycocalyx are carbohydrates bonded to proteins and lipids in the plasma membrane, and they are found in animal cells. So, plasma membrane, or the cytoplasmic membrane, similar in structure to prokaryotic cell membranes. It consists of a phospholipid bilayer, and it is important and integral, and peripheral proteins are present throughout. Integral proteins being internal and peripheral being on the peripheries. There are differences in structure, such as there are sterols, which are complex lipids, and carbohydrates, which are used for attachment, and cell-to-cell recognition and communication. Now, similar in function to prokaryotic cells, eukaryotic cells membranes have selective permeability, so they'll only allow certain substances to pass through. And they do have simple diffusion, facilitated diffusion, osmosis, as well as active transport. But differences from prokaryotic cells is that they can participate in endocytosis, basically phagocytosis and pinocytosis, the engulfing of material or the pinching off of material. Phagocytosis involves pseudopods which extend and engulf the particles, and pinocytosis is where the membrane folds inward, bringing in fluid and dissolved substances, and basically pinches off. Now the cytoplasm, that's still just the internal liquid of a cell. It's the substance inside the plasma and outside of the nucleus of eukaryotic cells. The cytosol is essentially the fluid portion of the cytoplasm, whereas the cytoskeleton is what the microfilaments form, and as well as intermediate filaments, and they give shape and support to the actual cytoplasm. Now there is cytoplasmic streaming, which is the movement of cytoplasm throughout the cell, allowing transport of materials. Like prokaryotic cells, eukaryotic cells do still have ribosomes, which are used for creating proteins. That's essentially what their main focus is. Ribosomes help make proteins. Now getting into organelles. The main one is the nucleus of eukaryotic cells. This is where DNA is kept to help protect it from either, say, radiation or chemicals, things that would mutate that DNA and possibly make it non-functional. The nucleus has a double membrane that acts as a nuclear envelope that contains the cell's DNA. And DNA itself is complexed or basically wrapped around histone proteins to form a complex called chromatin. And during mitosis and meiosis, basically mitosis, which is regular cell division, and meiosis, which is used in sex cells, the chromatin condenses and forms our characteristic chromosomes, our sectioned off sections of DNA. There are pores in the nuclear envelope, which essentially allows material to leave. In this case, allows ribonucleic acid to leave, or components of RNA to enter in, as the DNA is used as a template to form RNA. Now there is another organelle, which is around the outside of the nucleus. This is the endoplasmic reticulum. It's a folded network that consists of a rough and smooth portions. The rough endoplasmic reticulum is essentially studded with ribosomes, and these help form proteins. And then there's the smoother endoplasmic reticulum, which has no ribosomes, and it helps synthesizes cell membranes, fats, and hormones. Now the Golgi complex, or also called the Golgi apparatus, this is a transport organelle. It modifies proteins that are first made by the endoplasmic reticulum and helps package them and sort them to where they need to go, or add certain modifications as necessary. It will package those proteins in secretory vesicles, which can then go to the plasma membrane. Now there are mitochondria. This is where ATP is formed, ATP being the energy molecule that helps active transport. It consists of double membrane and contains many inner folds called cristae, as well as a fluid matrix. The inner folds add a lot of surface area to essentially allow for a lot of ATP production. And then also there are lysosomes, which are vesicles that are formed by the Golgi complex or Golgi apparatus, and they contain digestive enzymes to help get rid of, say, no longer functioning parts of a cell. And then vacuoles, which are cavities in the cell formed by the Golgi, and these basically bring food into the cell and provide shape and storage. Now, when we're talking about plant cells, specifically, there are chloroplasts. It's where photosynthesis happens. The conversion of carbon dioxide and water to sugar and oxygen thanks to the energy from light. And essentially they contain flattened membranes called thylakoids that contain the green pigment chlorophyll. Now other organelles are peroxisomes, which contain oxidized fatty acids that are used to help destroy hydrogen peroxide. There are also centrosomes that are a network of protein fibers and centrioles, and they form the mitotic spindle that is critical during cell division to move chromosomes to certain parts of a cell as the cell is splitting apart into two new cells. Now, the evolution of eukaryotes. Well, life arose as simple organisms. Estimates say 3.5 to 4 billion years ago. Quite a long time. And the first eukaryotes likely evolved around 2.5 billion years ago. Here, the endosymbiotic theory is essentially cells that came together. There is a larger bacterial cells that engulfed smaller bacterial cells, which developed the first eukaryotes. Now, it's believed that ingested photosynthetic bacteria are what became chloroplasts in plant cells, and ingested aerobic bacteria are what became the mitochondria in all cells, in all eukaryotic cells. A reason for this theory is because chloroplasts and mitochondria have their own set of DNA, whereas regular cells have DNA only in their nucleus. These organelles in particular, chloroplasts and mitochondria, they're very unique. And also goes into why your chromosomes, that part of your DNA, is half from your biological father and half from your biological mother, but your mitochondrial DNA only comes from your mother. Model the origin of eukaryotes, so essentially endosymbiotic phagocytosis, or endosymbiotic theory. Essentially, an early cell engulfed other types of cells that were much smaller, and well, the DNA eventually became evolved over time and gained its own envelope, which is where we have eukaryotic cells. And then eventually those eukaryotic cells began to engulf other smaller cells to form what are now chloroplasts in plants and mitochondria in all eukaryotic cells. And that concludes, essentially, Chapter 4 of this venture into online microbiology. Catch you all in the next lesson video. Peace.